بسم الله الرحمن الرحيم.
Good morning to all students of class 8. We are here with a new topic, and this topic is from chapter number 9, Reproduction in Plants. And we have done the vegetative reproduction in the previous class. We are going to recall the definition of the vegetative reproduction. Vegetative reproduction is the production of new individual from body parts like leaves, stem and roots. In this way, which is vegetative reproduction, in the vegetative reproduction we take the part of the plant, parent plant, and we grow that part, and from that part the same plant grow, and this is called vegetative reproduction. And we have discussed a few methods of vegetative reproduction. Today we are going to discuss artificial methods of vegetative reproduction. So we are going to discuss the topics layering, grafting, cutting, budding, spore formation, fragmentation, and binary fission. And we will see the advantages of the vegetative reproduction and disadvantages of the vegetative reproduction as well. So let's start from layering. These are different methods which are used as an artificial methods of vegetative reproduction. So in the layering, the part of the plant which are bent towards the soil, they are they are like covered. They are the some part of that part which is bent towards the soil, that is covered with the soft soil, and within few days the roots grow from that part, and then this part is separated and plant, and it grow into a new plant like the parent plant. So let's see from your book. In case of layering, the lower branch of the plant is bent down and covered under a thin layer of moist soil. Why need moist soil? Because moisture need to grow the roots from that part. Soil by pushing the tip into the soft ground. Now one part is covered with the soft moisture soil. After some time, roots develop and branch, and the branches forms a new plant. This is known as layering, because we have used one layer of the parent plant and we have grown it separately. When it grow the roots, then we will separate it and will grow as a separate plant. So this method is called layering. These plantlets can be separated out and can be grown as a separate plant. Such a method of reproduction is followed by jasmine and bougainvillea. These are the names of the plants in which this method can be used. Next method is grafting. Grafting, for example, there is a parent plant and we grow a new branch of another plant. A new branch of another plant on the parent plant means two different plants. There are two different plants, and the parent plant will called as stalk, and the plant new plant which we want to grow on the another plant, this part will be called as scion. This part will be called as scion. We take the branch, cut it down, and the stalk, which is the parent plant branch, we cut that branch and we just put the scion, which is another plant, and we place that on the stalk and covered it with the soil. So after few days, they are connected with each other. They are joined with each other, and all the nutrients which are necessary for scion, these are given by the stalk. So after few days, they bind firmly together, and then it new branch of a scion, which is desired plant, which it will grow on the stalk, which is a undesired, means we can grow on one plant the branch of another plant. So let's see from your book. A small branch of a plant is tied with the stem of the rooted plant of the same type. The joint is covered by a layer of wax or clay. We join it with the wax or clay. After a few days, the two becomes united and develop into a new plant. The rooted plant, which is already grown in the soil, The rooted plant with the root system under the soil is called stalk. This part will be called as stalk, and the stem cutting of another desired plant is known as scion. This is known as scion. The cells of the stem of the scion and the stalk connect with each other rapidly as if they belong to the same plant. This is facilitated by the stalk that supplies the desired nutrients to the scion. This method is used to grow mango, lemon, rose plant. These are the examples by which we can use the method of grafting and grow the desired one. It is very useful method as it is different to grow these plants from seeds or cutting, and also as it can give us desired varieties. For example, why we use this method? For example, if we take the example of mango, one plant is giving a very big, fleshy and sweet mango, while the other is not producing the good variety of the mango. Then we will use the good one, the branch of the good one mango, and will place it on the mango which is not producing the good variety, so we can get the good variety from both the plants. So in this way, these methods are used to get the desired varieties of the fruits. Let's see towards the cutting. Cutting, we have discussed in the previous class that we take a short stem of the rose plant, we cut it and grow it. So this method is called cutting. This is very common method to grow rose plant. A small cutting of the stem, a stem piece is taken and placed in the moist soil. After a few days, this stem gives rise to roots and forms a new plant. So this method is used to grow different rose plants. This method is known as cutting. Let's see the advantages of the vegetative reproduction. What are the advantages of vegetative reproduction? Why it is necessary to grow the plants by vegetative reproduction? Number one, some of the plants such as bananas do not produce from seeds at all. Banana do not produce seeds. So how we can grow the banana plant? The vegetative reproduction method is used to grow banana plant. They multiply only vegetatively. They are not producing seeds. They cannot be grown by seeds. They are just produced by vegetative methods. Number two, vegetative reproduction is faster. Vegetative reproduction is faster. It does not involve waiting for the seeds to grow, because when we grow the seeds, they take time to grow, and the smaller plant, then it grow, and after a long time they produce the fruits. While if we use some other methods, for example layering, grafting, cutting, so already the plant is grown, and on that plant, from that plant we can grow another plant. So we will not have to wait for a long time. So it is faster method. Number three, sometimes the seeds produced are not viable or fit. Sometimes the plants are producing seeds, but these seeds are not fit to grow a new plants. So for such plants, we can use the vegetative method. So that means they cannot, what do you mean by viable? Viable means they cannot form new plants. Means they are producing the seeds, but these seeds are not eligible to produce a new plant. So in this case, we can produce the new plant by using the vegetative reproductive methods. In such cases, the new plants are produced through vegetative means. Now come towards the page number 112. The next point which is important, that we are using the vegetative reproductive method, it is the embryo of the seeds that forms a new plant. It is the embryo of the seed which forms a new plant. If the embryo is not there or is destroyed, if the embryo is not there or destroyed, a new plant cannot be formed. So in this way, we can use the method vegetative reproductive methods. A new plant formed. Next is a new plant formed through vegetative reproduction are exactly like the parent plant. They are totally, completely look like the parent plants. They are like photocopies of the original plants. If the plants with similar qualities as that of the parent are required, vegetative propagation is the best method. So if we want that the exact properties which the parent plant have, then we will use the vegetative methods. Disadvantages of the vegetative reproduction. Since vegetative reproduction produces an exact copy of the parent, some undesired qualities of the parent plant also pass on to the plant of new generation. Means as they are producing the same copies of the parent plant, so sometime the undesired qualities, if any plant has the quality which we don't want, even then the new plants will produce, because they are having all the qualities like the parent plant, like they are the photocopies of the parent plant. So if there is any undesired quality, so we will have to face that quality in the new growing plants. Another method is the budding. So what is budding? Budding means the plants which are growing, they produce a small bud, a small portion on the plant, and it grow into a new plant. It grow into a new plant. If we will separate that bud and grow it separately, even then it can produce into a new plant. This is another type of asexual method. Sexual reproduction and asexual reproduction. Asexual reproduction, there is no need of male and female plants. Only from one single plant, a new plant can be produced. So this is called asexual, while in sexual reproduction, there are male and female plants are involved, and then the new plants can be grown. So all these methods which we are watching here, which we are discussing here, they are asexual reproduction method. This is another type of asexual method of reproduction commonly observed in yeast. Yeast, in the production of the yeast, the budding is involved. In yeast cell, a small rounded prominent part or a bulb-like projection is called bulb. Bud. What is bud? In the yeast cell, a small rounded prominent part or a bulb-like projection means a projection which is appearing on the parent plant. This will be called as bud. Start to develop in a side by the body. It soon enlarges in size, and then the nucleus, the large floating body generally in the center of the cell, divides into two. Then the nucleus divides into two. When the nucleus divides, it means a new cell is produced. One of these nuclei passes into the bud. Sometimes the bud at this stage separates itself from the parent cells to grow independently, and sometimes this bud is separated automatically, and it is grown as a new plant. And while at other instance, it may remain attached and form a colony of the cells, which refer to a collection of the cell joined together and producing more of their kinds. Sometimes they are separated from the plant and they are grown as a independent plant, while in the other way they are produced on the plant as a colony and they produce new plant from there. In a similar manner, sponges and corals form colonies. Budding takes place only when conditions are favorable for an organism to grow. When the conditions are favorable conditions are the conditions in which the plant may grow easily. There is a proper moisture, there is a proper light, there is a proper food. These are the conditions which are necessary for the plant to grow. So when these type of plants have the desired or favorable conditions, then they grow into a new plants. So bodies covered by a hard while if the conditions are not favorable, then the bodies covered by a hard protective coat that help them to survive harsh temperatures and lack of food and water. In the spore formation, we are going to start. Next is spore formation. Spore formation. A large number of fungi and bacteria such as yeast and bread molds. The example of the spore formation are the yeast and the bread mold. You have seen if the bread is kept outside and the conditions like the temperature is warm and when it get the moisture, some fluffy objects are grown on the surface of the bread. These are cotton-like substances which are grown at the substrate. Substrate will be the bread, the portion or part or the substance on which something grow. That will be called as substrate. So bread will be the substrate. They grow when the conditions are favorable and they produce the spores in capsules. These spores these capsules having the spores inside them, and when these spores burst, when these capsules burst, these spores again produce the new one. Okay, so in this way they grow. But if the conditions are not favorable, these capsules remain covered in the hard covering, hard protective coat, and when it get the favorable conditions, then this coat is burst and the spores come out, and these spores changed or grow into new one. Okay, so in large number of fungi and the bacteria such as yeast and bread molds, spores are produced from the parent body and germinate to give rise to young ones once the conditions are favorable. They can grow if the conditions are favorable. These spores are microscopic bodies. These spores are microscopic. These spores are very smaller that we can even not see. They look like the dust particles. Covered by a hard protective coat that help them to survive harsh temperature and lack of food and water. Spores represents a kind of resting state. In this way, the spores show the resting state when they are protected by a hard outer covering. But if the conditions become favorable, then they just grow into new ones. As soon as the condition turns favorable, spores break open and release the cells to grow and reproduce normally. Next is fragmentation. Certain algae and Spirogyra have a long ribbon-like filaments. These are present at page number 113, fragmentation of Spirogyra. These are ribbon-like structures. They are very long, and when we take a small portions, these will be called as fragments. And when these fragments are grown into new one, this process will be called as fragmentation. Certain algae like Spirogyra have a long ribbon-like filamentous body. It breaks up into two or more parts called fragments. The breaking of the body into two or more parts is called fragmentation. Each fragment grow into a new individual. So these fragments grow into new individual. Last and final is binary fission. Binary fission, binary means two. Mono means one, bi means two. So binary fission means from 1 into 2. Means one cell divided into 2. This is called binary fission. So how the binary fission takes place? When a cell grow at a certain point, then the nucleus first divide into it. When it make two nuclei, then the cytoplasm start to divide, and everything is divided into two parts. So from one single cell, two new cells are produced, and they separate. This process of separation is called cleavage, and by cleavage, one cell is divided into two cells first, but one nucleus is divided, and then the cytoplasm is divides. Binary fission is a splitting of a mature cell into two cells of the same type. It is most common method of asexual reproduction in bacteria. In binary fission, first the nucleus divide into two nuclei, then the cytoplasm divide into two parts, each containing a nucleus. Further stretching leads to the formation of two daughter cells. From the one parent cell, two daughter nuclei are formed. These cells grow into mature cells and then undergoes similar binary fission. So here amoeba, one amoeba is divided into two and different stages of binary fission, which are A, B, C, D and E. They are divided. Here just see. It's a one complete amoeba. Now the nucleus, which is in blue color, it is start dividing into two. Now completely the nucleus is divided, and here it is almost going to complete, and two nuclei, two daughter cells are formed from a parent cell. So here our today's class is finished. So let's come towards the homework. For the homework, it is present at page number 120, assessment 2. Assessment 2, B part, and question number B part, question number 3, and C part, question number 1. So you will do these questions. B part, question number 3 is explain the process of grafting in plants. Give any one example. And C part, question number 1, what are the advantages and disadvantages of vegetative reproduction? We have discussed the advantages and disadvantages. You will write it as it is. So do a very neat and clean homework, and take care. Allah Hafiz till the next